Welcome everybody to the Guy Foundation 2025 Autumn Series on Light. This is the fourth session in the series and we welcomed Glen Jeffery as our speaker today. To introduce him, we will have an introductory talk from Dr. Ifigeneia Kalampouka. Ifi holds a PhD in quantum biology and mitochondrial metabolism from the University of Westminster. The Guy Foundation funded her PhD and we're very proud of her achievements. Ifi is currently a business analyst in clinical trial AI automation, and she's a visiting researcher at the University of Westminster. Her research specialises in photobiomodulation, exploring how near-infrared light affects cellular and mitochondrial function, with a focus on mitochondria, ageing, and cancer. Ifi, we'd like to hand over to you now for the introductory talk. Thank you.
Thank you so much, Nina, for the introduction. Hello, everyone. I am Ifigeneia Kalampouka, and today I'm pleased to introduce Professor Glen Jeffery. His work establishes a central unifying thesis. Modern life, dominated by spectrally limited artificial lighting, has created a form of light malnutrition, depriving human biology of essential long wavelength light, specifically in the red and near-infrared light spectrums. So we are constantly exposed to blue light with the absence of longer wavelengths, as compared to what we could have gotten outside. This deprivation has significant and previously underappreciated consequences for human health and aging process. The core of Professor Jeffery's research explores the mechanism by which this occurs. The fundamental biological process and the basis of his work is the interaction between specific wavelengths of light and mitochondria. But let's take a step back and please let me properly introduce Professor Glen Jeffery before we dive deeper into his research. Professor Jeffery earned his PhD from the University of Oxford in 1982. He currently serves as a professor of neuroscience at the UCL Institute of Ophthalmology here in London. His institutional affiliation with Moorfields Eye Hospital of London is strategic. The eye and retina serve as powerful models for studying aging. The retina has the highest concentration of mitochondria of any other tissue in the body and ages faster than any other organs, providing an accelerated model of the aging process. And his mission is to transform the clinical and built environment through therapeutic light. Professor Jeffery's expertise spans neurosciences, ophthalmology, age-related macular degeneration, retinal structure and development, medical physiology, and the visual areas of the brain. His research on photobiomodulation appears in high-impact journals, including Scientific Reports and Aging Cell. And as we mentioned before, his strategic focus is the retina. As we said, it has the highest concentration of mitochondria, thus an essential tool of studying the mechanism and effect of longer wavelengths. Professor Jeffery has an academic and research network that is quite unique and multidisciplinary. As a quantum biology audience here in the Guy Foundation series, we won't be surprised that a professor in neuroscience not only collaborates with research leaders in medicine and different fields of biology, but also with physicists. However, what is truly unique about Glen's approach is his collaborations with architects and light engineers. Beyond academic publishing, Professor Jeffery has made extensive efforts in public science communication. He's appeared on at least 8 distinct podcasts, such as Sunlight Matters and Wise Athletes. And his YouTube presence is equally significant, with a lot of lecture and interviews across multiple channels. Now, let's examine the core science. Professor Jeffery's work demonstrates that mitochondrial function is central to health and longevity and is influenced by the spectrum of ambient light. The mechanism works like this. With red to near-infrared light, when red to near-infrared light strikes mitochondria, it enhances electron transport efficiency, increases the mitochondrial membrane potential, and allows mitochondria to produce ATP more efficiently. But here's the critical dichotomy. While long wavelengths are restorative, short wavelengths in the blue spectrum have the opposite effect. And timing matters crucially. The mitochondria appear to have a specific therapeutic window tied to their circadian work pattern. Professor Glen Jeffery has run quite a few important human trials, which he will present in a few moments. These studies have been crucial in our understanding of how longer wavelengths can improve age-related vision decline and can also have systemic effects throughout the whole body. However, what I can talk about today is the human intervention study happening in the University of Westminster here in London. After Glen examined my viva, we were talking and we started collaborating on a study examining systemic effects on humans. So Glen gave me an 850 nanometer light and we agreed on exposure protocol, 15 minutes every morning for 7 days. I then examined systemic effects in healthy volunteers over the age of 30. The two main parts examined were extracellular vesicles, EVs, and grip strength performance. I will explain on my next slide why EVs, extracellular vesicles, are particularly important, but briefly, they are membrane-bound structures released by cells into the extracellular space, and they can carry information all around the body for long-distance cellular communication. Grip strength, on the other hand, provides a functional measure of overall health and whole body mitochondrial performance, as muscle contraction is heavily dependent on that ATP production. Looking deeper on what extracellular vesicles are, EV carry important information from one cell to the other, and they can travel long distances throughout the bloodstream. Depending on the organism state, for example, stressed or not stressed, healthy, ill, inflamed or not so inflamed, EVs demonstrate specific changes. First, we have concentration changes. EV levels often increase in inflammatory conditions, cancer, and also neurodegenerative diseases. Second, we have size and content alterations. For example, the smaller size EVs, we call these exosomes, are massively released during stress and inflammation. EVs can also have pathological roles, but they also have therapeutic applications. Overall, EVs are crucial biomarkers and they offer insights into cellular communication. So, in this study, after isolating blood from participants before and after 1 week of 850 nanometer treatment, I first compared the EV concentrations before and after the intervention. Grip strength has been proven before to improve after near-infrared light effect. For example, this is a very interesting study that happened in Brazil in 2024. Nito and colleagues conducted a randomized, triple-blind, placebo-controlled trial with 60 intensive care ICU patients, applying photobiomodulations of 635 and 880 nanometers to critically ill patients. The results were very interesting. A 30% reduction in ICU length of stay, a 34% improvement in grip strength, and a very high improvement in mobility compared to the placebo group. This demonstrates that photobiomodulation can produce clinically significant functional improvements with even bigger effects in metabolically compromised patients. And indeed, we notice stronger effects in studies using diseased populations, where mitochondria likely have more room to become more efficient and are not functioning at maximum capacity. Photobiomodulation in 30 COVID-19 patients resulted in reduced hospital stay from 12 to 8 days, improved cardiopulmonary functions, and reductions in markers of systemic inflammation. Another study on long COVID patients this time used two different PBM approaches, a helmet targeting the brain and a whole body light bed. Remarkably, they saw cognitive improvements on validated tests. And although their initial hypothesis was that the helmet will have higher effect on cognitive improvement, actually, both exposure techniques have similar performances, demonstrating the systemic effect of longer wavelength lights. And the systemic nature of these effects opens therapeutic possibilities for metabolic disorders, neurodegenerative diseases. As we said, there are applications in the critical care settings and, of course, applications in the field of aging. And the key advantages are quite there. Bringing back long wavelengths to our bodies is non-invasive, low cost, it presents minimal risk, and it requires minimal logistical infrastructure. And taking into account all these benefits, the use of the full spectrum in our lives, or at least bringing back the red and near-infrared light, is a no-brainer. And I know there have been very important steps, including regulatory ones at government levels, but the most appropriate person to discuss all of these developments is a very key person behind the scenes and in front of the scenes in a few occasions, driving this science forward, Professor Glen Jeffery. Thank you.
Thank you very much. Great invitation to talk at the Guy Foundation. Great collection of people. I'd like to talk about light, mitochondria and health, and the title is Light, Mitochondria and Health, 2 billion years of evolution under sunlight until 1995. Okay, so let me give a very simple introduction to what I'm talking about, and although most of you will know about mitochondria. So early in evolution, cells generally lacked energy, and the world was fundamentally a place that was little more than green slime with not much happening. But then we think something very similar to a bacteria came along to a cell and said to it, look, if you let me in, I'll provide you with energy. You let me in and give me a home and give me a safe place to be, and I'll provide you with energy. And that's what happened. That became what we now call a mitochondria. And the consequence of that was cells had more energy, evolution took off, and we went from a green slime to complex systems. But the stranger, really, it wasn't a great deal, because the stranger came in, gave energy, but then introduced the idea of aging and introduced the idea of death, really. And that's the essence of a really great theory of aging, one that started in 1956, but still has a lot of traction. It was presented by Harman, and it's called the mitochondrial theory of aging. That mitochondria came into the cell, and the cell started aging and eventually died. So mitochondria, how do we think about them? Well, I'm not really a biologist by background, so I've got to generate constructions that are simple for me to understand and hopefully relatively simple for me to tell people about. So mitochondria are basically batteries. They make something called adenosine triphosphate, which is your petrol. It's the energy that you use moving muscles, kicking your legs out of bed in the morning, etc. ATP. We make our own body weight in it each day. It's a vast process. And the retina and the brain uses enormous amounts of this. The retina uses more of this than any other part of your body. But batteries fundamentally have a problem, and batteries run down. And in this case, our battery runs down with age and disease. And as the battery runs down, the probability of certain diseases increases as we age. So macular degeneration, dementia, Parkinson's disease, all increase with age and are associated with a failing battery. So we can think about our batteries controlling lifespan. Critically, the mitochondrial battery is influenced by light, and that's where I want to focus most of my argument today and talk about this organelle that regulates our life, but we can influence it because it's watching what's out there and it's responding to light. So mitochondria respond to light, but not evenly. So what we might call blue and red have a critical balance in environmental light, but mitochondria respond to them differentially. Okay, so longer wavelengths of light, here 660 to maybe 900. 900 is beyond our visual sensitivity. It's near infrared. Our visual sensitivity stops around 700, 715 nanometers. Those wavelengths increase membrane potential and ATP production. That means the battery has more charge and it produces more petrol. This is absorbed, we think, by the copper heme, a certain part of mitochondria. in cytochrome C. And what that does is it overcomes the electrical resistance on the battery. So to put it in more technical terms, we think that each photon, long wave photon that enters the mitochondria provides it with around 1 electron volt. But there is a resistance along the membrane of just over 1 electron volt. So by reducing the resistance along the membrane with a photon, long wavelength photon, we can promote health, and I will also show we can increase lifespan. Now, the trouble is that this balance, of course, has another side to it. Shorter wavelengths of light, 400 to 450, which is blue slipping into violet, reduces the membrane potential, tends to discharge the battery, reduces the petrol, the ATP. And this, we think, is absorbed by something called porphyrin in mitochondria, and it results in the production of oxygen singlets, which drive inflammation. And remember, aging and ultimately death is very often a result of progressive inflammation. And what I'm going to talk about, the experiments I'm going to talk about, they're identical in fly, human, and mouse. So I'm going to slip between them when I know that there is very strong evidence for a similarity. So these are highly conserved evolutionary mechanisms. Okay, so I've argued that light drives evolution for over 4 billion years, and light has a terribly wide spectrum. You can see another representation of it down here, which is sunlight, and it runs from just over 200 nanometers out to, in reality, about 3,500 nanometers. Although we only see 400 to 700, so we see a very, very small amount of what the sun is pushing in our direction. And we've been exposed to that wave light, us as humans, for over 3 million years in Africa, near the equator, on a 12-12 light dark cycle. We haven't had any clothes on for most of the time. And so we've got vast amounts of sunlight. But here's the snag. The snag is that as we progressed and developed, we moved into the built environment. And as we moved into the built environment, we had fires. That's okay, because fires produce the same spectrum as sunlight. Then we had incandescent light bulbs, those ones you put your hand on and they were very hot. That was fine because they had the same spectral output again as sunlight. Then at the end of the 1990s, beginning of the 2000s, people won the Nobel Prize for production of the LED. And the LED got rid of all the light that we don't see from around 700 all the way out to the end of the solar spectrum. Great, we saved money. Our lighting became much, much more energy efficient because our light sources didn't get hot, but there was a price to pay for that. So sunlight, solar light. What we've done here is an experiment to see what is our reaction to solar light. And we've actually taken a group of people from the Institute of Ophthalmology and we've stood them facing sunlight. And what we've done there is we put a radiometer and a spectrometer on their body so we can measure the wavelengths of light that are going through their body, and we can measure the energy that's going through. And I think, surprise to many people, we found that very long wavelengths of sunlight that are absent in LED lighting pass through the body. So here is a spectrum of sunlight as seen at the back of the Institute of Ophthalmology in London. And here are the wavelengths that we see passing through the body and coming out the other side. Be careful here because the axis here is very different from the axis on the other side. So the amount of light that we're getting coming through the body is very small, but that means the rest of it's absorbed. Here's a photograph taken with an infrared camera, very sensitive infrared camera, which is of someone's chest when they're standing with their back to sunlight. And you can actually see something. You can see some hairs on the chest. So the light is definitely coming through. We have to remember that infrared light, these longer wavelengths of light that pass through our body, can also be produced by plant matter. So all plant matter reflects infrared light. So if you take a photograph like this one with an infrared camera, what you see is all plant matter glowing brightly. And if you go out into sunlight on a very hot day and you grab a plant, you grab a leaf, it's not hot. It should be hot if it was absorbing the infrared, but in actual fact, it's cool. And it's cool because it represents a secondary source of infrared light. So you can walk out in sunlight, that's absolutely great. But also if you walk out in sunlight and you walk near a plant or a big tree like this, you'll get even more of it reflected back towards you. And just as a reminder for the difficult situations we find ourselves in now, you can see this is the spectrum of an LED, and there is that horrible blue spike, and there is an absence of infrared light from here. So why is this important? Well, I'm showing you that infrared light penetrates deeply into the body, into the liver, the heart, the brain. And I'm also arguing that these drive metabolism in a positive manner. So let's see about what other barriers we've got here. I'm going to talk about 850 nanometer light, which we can't see because that was around the peak that was passing through the human body. And here we have a picture of Bob Fosbury's hand, and his hand is sitting over an 850 nanometer light source. Of course, this photograph has been taken with an infrared camera, a sensitive one. Normally we can't see anything, but with the infrared camera you can see two things. Firstly, the light passes through the hand and you cannot see the bones. These long wavelengths of light are not limited by bone. So think about that in terms of your brain and sunlight. When you're walking out in sunlight, those long wavelengths are going through your skull relatively unhindered and into your brain. The other thing that's interesting is you can see in Bob's hand that the deoxygenated blood, the veins stand out and they are absorbing the longer wavelengths. Okay, that's the body. What about what most people do? Wear clothes. So here we've got some of Bob Fosbury's wardrobe. And it's 6 layers. It's a vest, it's a shirt, and it's a jumper. And this picture here is of that in an ordinary room with reflected light that we can see. This next picture is the same items of clothing, but with a light source that we can see behind it. It's within the visual range of sensitivity. The final picture is a picture with an 850 light source behind it. The point is that 850 goes through your body, comes out the other side, and your clothing acts as a minor barrier to it. So walking out in sunlight on a strong day, even if you've got plenty of clothing on, those longer wavelengths are getting into your body. Now, let's just set the scene very slightly about mitochondria. There are batteries. We've got to pay attention to some of their basic principles. And the first principle we need to think about is the fact that they don't stand still. They're not sitting there waiting for the world to happen. They need to know the time of day. If they're sensitive to light, they know the time of day. Now, mitochondria have complexes. They have different components that are responsible for passing the electron along the pathway to produce ATP. So we have complex 1, complex 2, complex 3, complex 4. We've measured the concentration of these complexes at different times of the day. So dawn, middle of the day, and the night. The interesting point is they're all moving around. So at different times of the day, you've got different concentrations of complexes. The mitochondria are detecting the light. The mitochondria are changing in response to the light. And let's look at our petrol. Let's look at ATP. ATP, the amount of petrol we make, changes radically over the day. It's very high in the mornings, and at night, our ATP production is relatively low. Now, we can produce ATP in another way, which is glycolysis, which is incredibly inefficient. And its byproducts are semi-toxic, not great for us, but we can see that we tend to slip over to this inefficient form of energy production during the night. Okay, so this is all shifting around, and I'm telling you that mitochondria see light, and I'm telling you that we can play with mitochondria optically. But let's look at when and how we can play with mitochondria optically. Let's shine red light at different times of the day. So this is the morning. We're looking at respiration here. This is a control, and this is when we use red light. So in the mornings, when our ATP production is very high, we can influence it. This is the afternoon. We can't do anything about changing mitochondrial respiration in the evenings or in the afternoons. So if you are going and paying for red light treatment, make sure that before you get your credit card out, that your treatment is in the morning, because if it's in the afternoon, you're wasting your time. Can I also point out something else, which is terribly important? A lot of our science starts with cells in dishes. We have cells in dishes, we grow them, we look at them, we take them out once a day or maybe even once a week, and we feed them. These cells have no idea what time of day it is. They're in a dark incubator, there is no light. When they are taken out, they're taken out and they're put into a hood, and they're put under a really offensive blue LED light. So as we progress through science, very often we do our first experiment in vitro like this, then we move on to a mouse, and then we move on to a human. If we're basing our science, particularly about mitochondria, on what happens inside an incubator, we are making a very fundamental mistake, because these guys have got no idea what the time of day is. Not only that, mitochondria have got highly complex interactions with all parts of the cell. So it's not just the mitochondria that get confused, it's the whole cell is in a very, very different and abnormal state in an incubator compared with what it's like in your body. A lot of science starts out in the dish, and we should be very wary of it. Okay, so a warning, but let's move back to the in vitro situation, and let's clearly see what happens when we turn that red light on. Well, if we look at respiration generally, so we're looking at the amount of mitochondrial activity. In this case, we've done it in flies. We take two populations and we look at their accumulative respiration, so how it increases over time. Two populations. One is a control population we just put on the bench. The other is a population we put into exposure of red light, and that is shown here. And you can see that from very early on, the two populations separate. So within a relatively few minutes, the red group of flies are separating from the controls. So from early on, that red light is influencing mitochondria and respiration within a few minutes. What is the consequence? The consequence is when we look at ATP in different parts of the animal, in this case, it's a mouse, we find we have more ATP in the retina, more ATP in the cortex of the brain, and more ATP deep in the brain in the thalamus. Remember, that red light is going through the skull. So we are perhaps, and this is the thought, that the red light is increasing the spin on the AT rotor pump to produce more ATP. Possibly it's doing it via reduction of the viscosity of the water around the ATP pump. That's been something that's been talked about, because nano water is highly viscous, but something else happens, and that is we start producing more mitochondrial proteins. Cytochrome C oxidase, a key protein in respiration, is increased. And we can see that very clearly here and here. So my analogy is with grommet. When we give the red light, the first thing that happens is grommet moves faster on the train. But after a period of time, that increase in ATP results in grommet laying new tracks. So we increase ATP production, perhaps by increasing the spin rate of the ATP rotor pump, but then we reach a certain point and we say we need more mitochondrial proteins. We need to build more tracks. That's really important. So the first thing is the train moves faster. The second is the response to the train moving faster is to produce more mitochondrial proteins. What happens when we produce more mitochondrial proteins? We increase mitochondrial behavior, we increase ATP. Well, when you increase ATP in aged animals, you can impact on inflammation. Remember Harman's mitochondrial theory of aging. As mitochondria decline, you get a reduction in ATP and you get an increase in inflammation. So here we've got some aged mice and we looked in their retina. It's my territory, it's where I work. And we've looked at a whole range of markers after we've given them a burst of red light. So macrophages are a very, very clear indication of inflammation. When we give the red light, macrophage numbers go down significantly. Another clear indication of inflammation is complement. Complement protein expression, and this is one of the complements, C3d, that's gone down significantly. C3b, that's gone down, but it's not quite significant. An independent marker of inflammation, independent from complement, is calcitonin, and yep, calcitonin's gone down very significantly. And one that we probably all recognize as an inflammatory marker, TNF-alpha. TNF-alpha has gone down as well. So consistent with the mitochondrial theory of aging, red light reduces inflammation. That's an important point for thinking about how we deal with the aging process with something that's really a very, very simple and straightforward technology. Okay, mitochondria also do something else. If you think back to the start of my talk, I said mitochondria regulate cell death. Consistent again with Harman's theory, mitochondrial theory of aging. Now in the retina burns very, very fast. It's got a super high metabolic rate, highest in the body. And the consequence of that is cell death is a key feature of aging, the aging retina. In fact, both us and mice lose about 30% of our central photoreceptors over life. Now, our lifespan originally was maybe only 35 to 40 years, and that was absolutely okay. But now the average life expectancy in Western Europe is around 82 years, and the consequence of that is that's not an environment our body is used to. So we've taken old mice, 8 months, we've taken old mice, and for 8 months daily, we've given them a burst of red light, and we've counted their photoreceptors, and we've done this in 2 independent ways. We've counted the nuclei of the photoreceptors, and then we've counted the outer segments of the photoreceptors. So quite a challenge, I can tell you, when you can see that these are the outer segments of photoreceptors. So first of all, here is our young animal. Here is an older animal, and there are far fewer nuclei in the older animal. It's gone through a process of cell death. If we give that animal a burst of red light every day, we have reduced the pace of aging. We have reduced cell death. If we now look at the outer segments, and it really is a horrendous task to count these, again, we can see that we've got here roughly 30% loss with age over time. And we can see that our treatment with red light has resulted in a reduction in the pace of cell death. So by giving long wavelength light, we improve mitochondrial function, we improve ATP production, we reduce inflammation, and we reduce cell death. I think these are important points in an aging population. Okay, can we watch this in real time? Yeah, actually we can. We can watch mitochondria breathe in real time because they do something that actually is quite unique. Or it's unique to me because I'm not a physicist, but engineers at UCL Biomed Engineering, Ilias Takhtanidis, has shown that mitochondria change color as they breathe, and that you can monitor this change in color to monitor how the cell is responding. So superficially, this looks really simple, but the software behind it is pretty impressive. So we shine a light into quite a dim light into a mouse eye, and we pick up the reflection that comes from it, and we look at the reflection in the wavelength range that we know mitochondria change their color in. And when we do that in a stable preparation, we can sit and we can watch, in this case, cytochrome C oxidase, a key component in mitochondria, and we have a stable preparation, and we give it a burst of red light for a few minutes, and the respiration changes very markedly. And in that way, this is non-invasive. We don't have to touch anything. We can do this with a small gap between our probes, our fiber optic probes, and the cornea of the eye. So we can watch mitochondria breathe without touching you. I think this was developed by Ilias Takhtanidis. He's the hero of this situation, but I think that's really impressive. And you can do it in skin, super easy. We can do the same with humans. In fact, we can do a lot better with humans. How do you stress the human retina? You do it really, really simply. You turn the lights off. If you turn the lights down, the thing that the retina responds by saying, I can't see anything. So it turns the gain up on the amplifier. It burns more energy to try and find the signal. One of the therapeutic approaches to some age-related eye diseases is actually to keep the lights on at night, and that's particularly the case in diabetic retinopathy. So let's look at a group response of young and old people. So there's going to be a difference in mitochondrial performance between those two, to turning the lights out in the reflected light from the retina. So as we turn the lights out, we find that the younger person basically improves their cytochrome C oxidase expression as they dark adapt to the environment. If we look at people over the age of 50, we know people over the age of 50 can't dark adapt properly. Normally, dark adaptation is about 35 minutes. Dark adaptation in older people is 50 minutes, and it doesn't plateau at anywhere near the same pace. And you can see here within 3 minutes, we have a clear difference between the two groups, the younger group and the older group, in their performance. Now, we can take an individual, in this case me, and I was definitely in this group here, not doing very well. And here is my performance, and that curve is part of that function there. I then went away, had a cup of coffee, a burst of red light, and came back and was repeated. And you can see that my mitochondrial response, my respiration in my retina, changed markedly as a consequence of exposure to longer wavelength light that we believe was linked to changes in my mitochondrial performance. How can we look at this maybe perhaps at a population level? As I've gone along my career, I realized that human psychophysics is a lot cheaper than working on animals, and people tend to read your papers when they feel they can be related to it. So can we improve human vision just by exposing people to different wavelengths of light? And what we've done here is I've presented letters on a very noisy background. and I've asked people, what's your threshold for detecting this? So if I have a letter A in blue on a very, very noisy background, what's your threshold? Where can you detect it? So in black here, you find the responses of people to that threshold. It tends to go up a little bit with age. So I ask them to identify the letter. I then give them 3 minutes of red light, and I bring them back, morning only. What happens? The red squares are all below the black dots. That means the threshold for detection has improved. Right now, the population level here, that improved to the tune of 17%. Some people notice it, some people don't notice it. And the effect, whether you're doing this in a fly, a bumblebee, or a human, lasts 5 days. So 3 minutes here buys you 5 days. And we find bigger effects for detection of blue than we do for other colors. And that's partly because you shouldn't really see blue. You've got very, very few short wavelength sensitive cones. They're very frail in aging, disease, and diabetes. So we can improve human vision by exposure to long wavelength light that improves mitochondria. Now, if he's talked about the issue of communication around the body, microvesicles, we found when we stuck people outside that around 850 nanometers was what we could measure coming through their bodies. So what we've done here is we've done an experiment where we've exposed light to people's back. And here we've got a ceiling panel with an array of 850 nanometer LEDs in it. And we've kindly asked them to come into the room, and we've exposed their backs, and we've tested their vision. Okay, we get an improvement in our thresholds. Our thresholds go down. But then what happens if we take a really big roll of kitchen film, we wrap their heads in it? We wrap their heads in it so they can't see anything. The eye cannot mediate this response. We get a reduction. Now, the reduction isn't as large, but in the case of blue, remember blue is pretty frail, we actually find it's a significant 7.5%. So I want to introduce the concept to you that mitochondria communicate across the body and they act as a community. You can do something to the skin and eventually cells in the retina get to hear about it. Now, John Metrofanis, who has done some absolutely outstanding work in ameliorating the impact of Parkinson's disease in primates, has done very similar experiments, very, very elegantly. You can induce Parkinson's disease overnight with a drug. So the animal wakes up and it's very rigid. It's very, very, yeah, it's got full-blown Parkinson's disease. Now you can put a probe into the brain, into the brain stem, and you can deliver red light and you get big positive changes. But what John did, which was really impressive, was he looked at controls. So he put red light on different parts of their bodies and found in each case an improvement of around 70% in the symptoms of those animals. They're a community. Now, if he's gone on and done a much better study than me, but this was our basic study because I started taking blood from mice saying, what's going on? What's going on in the blood when I give red light? Well, I kind of just grabbed stuff off the shelf. What have I got? So we had a lot of cytokine kits and we found that, horrible picture, I know, just look at the black and the red. The black is control, the red is experimental after red light, and that is a cytokine profile. Now, don't get particularly concerned about the fact that the red tends to be higher. The point is small increases in cytokines put the body on a protective stance. I think Ifi, with her microvesicles and the potential for their cargoes, will actually be telling us a lot more about this. And this happens relatively quickly. You can shine the light on the foot or on the abdomen, and you get a change in under 24 hours, an improvement. So I want to talk about how we can push this forward clinically. I also want to talk about what the dangers are of the short wavelength light, because it comes to dominate when red light is absent. We were doing lots of work on mitochondria, and I was using the words like mitochondria and improvement in papers. And I started to get emails from people saying, you know, my child has mitochondrial disease. I later discovered that this was an absolute, you know, in some cases a killer, because sometimes kids didn't make it beyond their 20s. Very often they died through heart failure. They have got typing errors in their mitochondrial DNA. Mitochondria have their own DNA. And the consequence is they have a reduction in the amount of ATP they produce. So I didn't have ethics for this. So I told people what I might do under the circumstances, but we didn't do a proper study. This was the first character that came along. She's very ill, not really spending much time outside bed. Her face has lost its symmetry because just her muscle control has gone. We were looking for ptosis, the ability to open the eye. Because we were looking for ptosis, we used a torch on the eye, and you can see the dates underneath. We used this here, and this is where we ended up. This young girl now, she could probably walk about 50 meters in the mornings. She walks to school, and that's more likely 800 meters. This chap was far less affected, but he did have ptosis, and we did improve the ptosis. So this was originally, we then got a grant from the Lilly Foundation, and we've been doing a lot more kids. The trouble is, A, we're not getting enough kids to do it, and B, these children, some cases, are really, really sick. So we've not been able to progress that. If we had a larger population, I'm sure we could. Even if it only worked in 2% of the kids, that's a winner. So bigger concepts here. Mitochondria regulate your lifespan. I can't do lifespan experiments with people. I can't do lifespan experiments with mice because UCL charged me, I think it's 43p a day for a mouse. But I can do it with flies. So if we take flies and we divide them into two groups and we count them every week, a fly will roughly live 12 weeks under laboratory conditions, and we give one group of those exposure to 670 for a brief period every morning, and the others, the controls, we do not, and we count them each day. The consequence is very clear. It's been repeated time after time here. The flies that get long wavelength light live longer. They live longer in relative terms because the middle age deaths are reduced. They don't live longer generally in absolute terms, but the middle age deaths are really eaten into. And these flies also climb better. So we've got longer lifespan and we've got better motor skills. They also do some really interesting things that I really want to carry on exploring, which is their cognitive ability. If you put a fly on an island and you take its wings off, the fly will do a standard series of things to try and find out where it is. A young fly will go round and round and round in circles. It wants to know the limits of its world. A 6-week-old fly, it really doesn't do it very well. But a 6-week-old fly, given an exposure to long wavelength light, he doesn't go back to being young, but he's a hell of a lot better. He's improved. Now, okay, locomotor function, we had that, but also their memory improves. So if you get a fly to run down towards a light down a tube, it'll do it instantly. If you then put inside that tube quinine, it gets to the quinine, it hates quinine, it goes, ah, and it doesn't go down the tube. You take a young fly and you put it back in that tube the following day, and it goes, I'm not going down that tube. I know there's quinine there. I remember that. So older flies don't. So we have a big reduction in memory function in those older flies. older flies that we treat with 670 nanometers, remember that there's quinine in the tube. So there is an improvement in memory. Now, if you're improving mitochondrial function, you're improving ATP. Where's that ATP going? It's going on membrane pumps. The vast majority of ATP is burnt on membrane pumps. And membrane pumps are throughout the central nervous system. So one would expect general improvements. And so this was a really nice result. Okay, a little bit of digression here because I was cycling home. It was pouring with rain, and I just read an article on neonicotinoids, which are insecticides that's specifically toxic to insects, fantastically cheap, and they induce Parkinson's disease, and the fly, the bee, the insect doesn't move and it starves. I thought, oh, this is great. We've got something that specifically hits mitochondria. Can we redeem them? This was a great experiment. First of all, because it brought bees into the lab, and bees are just the most fantastic animals because they remember, they learn, they do great things. So if we take a group of bees and we poison them with imidacloprid, they just don't live very long. If we give them imidacloprid and red light, we improve that situation very significantly. But the bees that we just give red light to are like the flies. They live longer. The middle-aged death has gone. And one of the sidelines of this is that in America, they've used a lot of insecticide and now have got a pollination problem, and they drive lorries with thousands of beehives up from Texas and Florida to Northern California. And when they bring them back, a lot of these hives are dead. But now they've got, some of them have got red light in them. And I understand that that's led to a significant improvement in survival. And these businesses are run by accountants, so they're super happy with that. We'll avoid that. So, okay, we can improve lifespan, we can improve health. This project was not my idea. It was actually an idea that came from my work actually on a long drive down to Porton Down for a series of projects we were doing there. And Mike Powner on that long drive said, if you can make mitochondria work harder, they should eat more sugar. If they eat more sugar, your blood glucose should go down. I thought that was too simple to be true. I won't bother you with the experiments we did on bees with that, with red light and blue light, and moving their blood sugars, their hemolymph up and down. But this is an experiment which you really need friends for, because you need to get people to drink a vast amount of glucose, which is disgusting. You have to prick their finger every few minutes to measure their blood glucose. And then you've got to stick a tube up their nose to measure their oxygen consumption. Because if mitochondria are improving, they should eat more glucose and they should produce more carbon dioxide. So you can imagine seeing the curve, the functions here, what happens. These are the normal groups of individuals. Their blood sugars spike around 40 to 60 minutes after consuming the glucose. When we give people a burst of red light on their backs that accounts for really around 10% of their body surface area, we get an increase in oxygen consumption and we get a decrease in blood sugar spiking. Now, these are healthy individuals. Mike Powner has just finished a type 2 diabetic study and he's got exactly the same result. So systemic changes, that's the big thing. We can get systemic changes with distal exposure of the body. Why is this so terribly important? Well, it's terribly important because this is a World Health Organization and it's basically problems with diabetes. Where is our problem with diabetes worse? It's worse in the Gulf. Why is it worse in the Gulf? It's worse in the Gulf because they never go out in daylight. You can't go out in daylight, you fry. So you go from your building with its horrible LED lighting straight into your air-conditioned car. And so we were really lucky we were invited out to Kuwait because they've just now got a really big institute basically just centered around the acknowledgement of diabetes as a big problem. So I want to move on to this more kind of bigger, wider picture And this is an experiment that Edward Barrett and I did. This is here east. It's a really horrible building. It's a terrible building. It's a leftover from the Olympic Games. It's miles away from absolutely anywhere. And it has infrared blocking glass and horrible LEDs inside it. So this picture here, Edward has a very sensitive infrared camera, and he goes around taking infrared pictures. And this is Edward taking a picture in infrared of the front of this building. It's a mirror. It's a mirror because it's reflecting all the solar infrared light. When we go in through the door and we photograph the inside, we find we can only see infrared when the door is open. And you can see there's some plants there and they glow. So infrared blocking glass is a major feature of modern architecture, right? So the LEDs are robbing you of some of your long wavelength light, and the architects are robbing you of the rest of it with infrared glass. So here is an experimental area that Edward introduced me to. It is where the architects make their toys. It's where they make their models. It's horrible. It's got really brutal light. Guys spend a lot of time in there. And the idea of the experiment was to walk in there and give them a really small amount of sunlight. And we did that with an incandescent light bulb, because incandescent light bulbs produce the same spectral output roughly as sunlight does. They've got acres of infrared light. So here's one of the workspaces, benches that they use. Here's Edward with his camera photographing it in infrared, and there's nothing there. And there's Edward photographing it again when we gave them fundamentally IKEA lamps with 40-watt light bulbs in it. A lot of infrared, and the infrared bounces around a lot. And we gave them those lights for 2 weeks. First of all, we went in, we measured their ability to detect color contrast, and we left them with the lights. These guys weren't sitting under these lights all the time. These lights were part of their work environment. They got up, went to the toilet, they got up and had lunch. What was the outcome? The outcome at the end of 2 weeks was a very significant improvement in their color vision, in both red and blue. We took the lights away. And then I said to him, well, we better go back and just see how it bounces back. So we went back at 4 weeks and we went back at 6 weeks. And the improvement remained even though the lights had gone. A big problem here was I wanted to go back continually until we got back to baseline. We were floored because this was Christmas. This was this time last year. This was Christmas. We couldn't carry on with the experiment because all these people were now going to change all their behavior. They were going to eat loads. They were going to drink loads, and they were going to sit around or go to relatives outside. So the incandescent light bulb, which you can still get, which has got vast amounts of infrared in it that you can't see, is having a very big effect on mitochondria and your color perception, both blue and red domains. If you remember, previously we could only get big improvements in the blue, less so in the red. What else is happening in our world which is LED dominated? Well, interestingly, NASA published a big paper in Cell. Cell's an important journal. And it basically said, we've got a mitochondrial problem with our astronauts. Had a large number of authors in it. You could almost feel the amount of money that have been spent on this project. That's their world. Their world is basically 24 hours a day, white LEDs. You can see it doesn't do much for your hair either. Mitochondria control the pace of aging. This was the lady who got stuck up in space for a year. She should have come back in a couple of months. Leave you to draw your own conclusions here, but the aging community, or people, the aging research community, have seen this, and they're paying a lot of attention. All of the astronauts tended to have shortened telomere lengths, but some suffered a hell of a lot more than others. Now, NASA didn't always respond as much as we would like to questions about this, because they want commercial flights to Mars, and no commercial company is going to take on a role like that if the outcome is going to be people age rapidly. But the LEDs do a lot more. If we take older mice and we put them under basically 420 or 450, those are the prominent peaks in LED lighting, you get fat. You get fat really quite quickly. We're slowing our mitochondria down, and as we slow our mitochondria down, there is more glucose remaining in the serum. So body weight changes quite a lot, but other things change as well. Where is the other biggest concentration of mitochondria? It's in sperm. So if we look at the sperm from animals that are kept under 420 or 450 nanometer light, the heads of the sperm are significantly deformed. Again, another sign of a systemic effect that's happening as a consequence of blue light exposure. And for the poor mice, I mean, this is a kind of a terrible scenario. They're older mice kept for a longer time under these wavelengths. Their hearts are lighter, their kidneys are smaller, their livers are smaller. They put on a lot of white fat. Liver size is also correlated with an increase in pathological liver function, which we measure generally with ALT. Their behavior changes. If we put a mouse in a box and then film how it behaves, well, it runs around the box in all parts. If you've got a mouse that's been exposed to these short wavelengths, its behavior becomes cautious. It only runs around the edges. And that is seen in animals that have low-level systemic inflammation. Having seen the image of the sperm of the mice, I asked this group to actually do the histology on the testicles. Here's the control, and here's that of that exposed to 420 or 450 nanometer light. So blue LEDs are undermining health. Short wavelength light now dominate the built environment, and we're now spending more than 90% of our time inside. This is a really big public health issue. Skin mitochondria detect short wavelengths and they signal it around the body. These wavelengths reduce lifespan and they increase stress. And we also know that they undermine insects. We've got loads and loads of environments now where we've got high power, short wavelength light. So floodlights, motorway lights, awful. Here's a child born who's, it's not uncommon at all, liver function hasn't quite kicked in, and we exposed the child to short wavelength lights break down bilirubin. Interestingly, we generally do this for about 2 days here, and I was rung by a nurse in America saying, why do we do it for 5 to 6 days in America? And the outcomes seem to be because you can charge more money for it. Mitochondrial decline is a feature of diabetes, Parkinson's disease, macular degeneration, dementia, etc. And it's key in aging, and we all suffer from that. And I think the point that I really want to make is we should be thinking about our lighting in our public environments. Many of our buildings are glass, and that glass is blocking infrared light. Here's our new Institute of Ophthalmology. It's almost topped out. It's all glass. The glass blocks infrared light, absolutely beautiful. Edward's been around there trying to photograph it, and it's going to have cheap LEDs. The thing to always remember is that in a building, the last thing that ever goes in is the lighting. If you've overspent on anything, any item whatsoever, it has to come out of the costs on the lighting. But we are getting a change here. We are denting things. So we've been able to make some changes to lighting in Queen Square in the critical care unit. We've been talking to critical care at King's College Hospital. Fantastic unit, absolutely fantastic. awful LEDs. Edward can't find a stray of infrared light in the building. So spectrum of light is very, very different. We are now living in a world with LEDs. We've got no infrared light. Our floodlights are absolutely awful. They have big blue spikes in them. And BMW have just produced, or relatively recently, they produced a laser light in their car. So those of you that have bought BMWs, they've got a killer of a short wavelength spike right in the right place. And the Daily Telegraph motoring correspondent said, I got caught in the beam of that and my eyes didn't feel right for a couple of days. I can understand that. So finally, reiterate what has been said previously. You live longer and suffer from fewer diseases if you spend a lot of time in sunlight. Now, Richard Weller brought this out. Richard's argument was very much it was UV. I'm arguing that a lot of it is long wavelength. I think from the point of view of public health, does it matter? I don't think it matters that much. We need to think about lighting. And I just want to thank a few people here. Bob Fosbury, who keeps telling me every time I say, I think I'm going to do that. He says, we know that because he's a physicist. Jamie, who was my postdoc, who worked for ages. Mike Powner, who got the stuff on blood sugars really well. Edward's been unbelievably helpful from environmental engineering and architecture. Ife, we know. Scott Zimmerman has also contributed enormously to our understanding and our discussions on this subject. Thank you very much.